Go ahead. Okay, hello everyone. My name is Grace Peng, and I am the chair of the Interagency Modeling and Analysis Group, IMAG, and I would like to welcome you to our IMAG Multiscale Modeling webinar series. And today's webinar is being hosted by the Cell to Macroscale Working Group, and one of the co-leads, Dr. Ed Sander, is hosting this webinar, and I will turn the mic over to Ed. Thank you.
Thanks, Grace. I'm Ed Sander. I'm co-lead along with Qinglong Lin of the Cell to Macroscale Working Group. Our working group seeks to try to tie together subcellular, cellular, and macroscale processes using modeling, mechanics, imaging techniques, a whole range of different things. We try to have 2 webinars a year, and I'm excited that our second webinar for the year will be by Dr. Knothe Tate, and so I'll turn it over to her.
Thank you so much for the introduction, Ed. Thank you so much to the working group, to IMAG, and to Grace for the invitation to present today. We're really excited to share with you our coupled seamless organ to multiscale imaging and modeling approach as a tool for discovery, biotech development, and diagnostics. This work is the result of a multi-year collaboration and continuing collaboration with Zeiss Microscopy and with Cleveland Clinic. And I'd like to acknowledge my co-presenters this morning, Dr. André Pereira, who's a postdoctoral fellow in my laboratory, and Dan Hagema, who's a doctoral student in my lab. Just before we get started, I would like to mention I'm the Paul Trainor Chair of Biomedical Engineering here in Australia, in Sydney. Paul Trainor was the founder of biomedical industry here in Australia. He was the inventor, and he commercialized the cochlear implant and pacemaker here. And a lot of the work I'm doing here is in the Trainor legacy. I'm trained as a biomedical engineer, both in mechanical engineering and in cell biology and biology. So a lot of the work that we do in our group, we call ourselves the MechBio team, to show that the work we do is at the interface between mechanics and biology. And you'll see today that our work really goes across these length scales and really continues this interface. I do have a few disclosures to make with regard to commercialization of our intellectual property and startup and spin-off companies that I have been involved in and continue to be involved in. Let's get started with the science and engineering. The human body is a complex biosystem, and we realize that our organs themselves, they have bounding barrier membranes. They're very much like ecosystems that are brought together for the complex organismal system of the human body. In recent years, we're realizing that our cells themselves, from in utero development through to postnatal growth and healing, are prototypers and inhabitants of this ecosystem. They're also beacons of health within this ecosystem. So we see this comparison here to the rainforest of the Amazon, and the cells or the trees in the forest actually give an indicator of health at any time. As I mentioned before, this developmental process starts in utero during development, and it's the cells themselves, which we often forget, actually build the tissues, and these tissues are adapted by the cells throughout life. An important concept in understanding the adaptation of our biosystem, our bodies, throughout life and in health and disease is that we understand the length scales and the time scales of this process. And if we have that knowledge and start to understand the mechanisms behind this, we'll have the capacity to develop smart materials that emulate the human body's own tissues, as well as new technologies and systems. And that's a lot of what we're doing in the MechBio team. And just for this comparison here, if we look at the length scales and the time scales between an individual osteocyte here in a human bone and a tree in the Amazon rainforest, the orders of magnitude of length scales are relatively similar. If we think of the rainforest, this is the lifespan of the rainforest goes over millennia, and just like the evolution of our skeleton has developed over millennia. An interesting aspect of this approach is that there are analogous paradigms across lengths and timescales, as well as across species, organs, and tissues. And recently we've recognized that this connectivity, for instance, here of these osteocytes in human bone, there's a similar paradigm in the brain here. You can see these are dendrites in the brain, and the connectivity gives an indicator of health status, as well as the connectivity decreases with age and disease. We can see that as a beacon or an indicator of incipient or progressing disease, here in bone, as you can see here, and here in brain with injury. These very small-scale phenomena then emerge as large-scale and organ-level disease or failure, as you can see here in the hip. And understanding and emulating of these processes is very important, as I mentioned before, for the development of smart materials and smart systems, which by nature adapt to dynamic stimuli in their environment. However, until recently, this idea of multi-scale computational modeling, there was the hype versus the promise, and until recently where we were able to start using really exciting technological development that enable the seamless imaging across all these length scales, we've had a really difficult time as modelers to bridge the length scales. So we see computational modeling as an enabling platform, not just for discovery, but also for validation.
I'll open the door. I'll open.
Top-down approach to modeling, as well as a bottom-up approach, where the top-down approach going this way is more environment-centric, and the bottom-up approach is more cell-centric. And these approaches have enabled the development of advanced materials, devices, and diagnostics. And if you're interested in more details on this, they're reviewed in some recent publications of ours. Let's get down to the multi-scale measures of this population connectivity. So if we think of these cells, they're the inhabitants of our tissues and organs. And it's very important to be able to measure the connectivity of these cells. One measure for that would be the permeability of this network. And one of the questions we are interested in is how we can measure this permeability, which would be a surrogate measure for signal transfer efficiency between the cells, and ultimately will enable us to predict the earliest onset of disease. So one way that we do this is that we take our microscopic, these 3D microscope images of the cellular networks, and we take the inverse of them, we scale them up using similitude theory, scale them up by 1,000 times, and we can actually measure permeability of surrogates of these cellular networks. But as you can notice, the field of view and the volume of interest here is really small. It's 159 microns on edge each way here with a shorter depth. And this enables us to start to make predictions at very short length scales. The other thing that we do is we build nano to microscopic fluid dynamics models to understand the flows around cells. And we've made great inroads at very small distances and length scales. With these models, we've realized that a very precise representation of the geometry and the microanatomy and the nanoanatomy is very important. But until recently, with our collaboration with SAIS, we haven't had the method to do such seamless imaging. And that's really what today's webinar is about, is these multi-scale approaches and the coupling of the imaging and the modeling. Just to reiterate this theme of using multi-scale approaches, much of our nano to microscale modeling has been carried on by a former protege of mine, Dr. Eric Anderson, who now is the chief fluid dynamicist of the National Oceanic Administration's Great Lakes flow region, and he's using the same types of methodologies for flows through the world's largest freshwater reservoir. So as I mentioned before, the cells themselves have a profound effect on tissue and organ health, and we want to be able to understand that at very long length scales, which are the length scales of relevance that we're thinking of.
All right, so now I'll show how we used a multi-beam scanning electron technology to develop a multi-scale imaging platform. So this paper shown here in the screen, this was accepted in PLOS Computational Biology, and it's coming out very, very soon. So for further details on the methodology, please refer to that publication. So a little bit of a background on scanning electron microscopy. A scanning electron microscope is a technology that produces images of a sample by scanning it with a focused beam of electrons. The electrons interact with the atoms in the sample, producing various signals that contain information about the sample's topography and composition. And it can resolve morphologic details at the nanometer scale, and is commonly used to characterize the structural and functional properties of biomaterials, biological tissues, and their cellular inhabitants. The acquisition of electron microscopes, however, limits the capturing of high-resolution images within reasonable time frames, and therefore, typically the throughput is limited to a scanning of areas within the micrometer range. So, multi-beam scanning electron microscopy, developed by Zeiss, which we're going to also refer here to as MSEM, circumvents the typical throughput limitations of conventional single beam SEM. By operating with multiple parallel beams, the second electrons that emanate from each primary electron spot are collected by the objective lens and imaged into a multi-detector device with a collection of channels. So the parallel electron beams in MSEM are arranged hexagonally to minimize electron-optical aberrations. For this particular sample, we used a 61-beam MSEM prototype, and therefore each multi-beam field of view was composed of 61 single image tiles arranged in the hexagonal pattern that we see on the image. So this microscope has drawn interest within the scientific and technology community, particularly in the assessment and detection of nanometer-sized defects in silicon wafers, in areas related to brain connectomics, and as we will see in this presentation, also in cross-scale musculoskeletal mechanobiology. So this is a bit of an overview of the acquisition process. Bone tissue samples were acquired from human patients undergoing hip replacement. We used the tissue normally in the course of surgery, and a section of the femoral neck was prepared according to techniques adapted from atomic force microscopy. We imaged roughly a 3 by 3 millimeter section, which resulted in about 900 multi-beam fields of view. Each field of view, as mentioned before, composed by 61 tiles. The whole image dataset in this case was comprised of about 55,000 high-resolution image tiles, and each image tile had about 10 nanometers of resolution, which allows to observe with detail cellular and tissue structures. So ultimately, if we want to image from the whole organ to its cellular inhabitants in a seamless way, we need a way to reconstruct this collection of tiles. So first we calculated the correct alignment for all pairs of overlapping images. Then we found the global positions of all tiles that comprise the data set. And finally, we developed a platform to visualize, navigate, and analyze these data sets. With alignment, motorized stages that I used for such acquisitions operate with quite a high precision. However, in most cases, the physical coordinates from the stage are not precise enough to provide a proper reconstruction of the whole action at the pixel level. So this means that the position of tiles created visible misalignments and therefore required post-processing. We use pixel-based alignments to register all pairs of overlapping tiles. And so this is a Fourier-based alignment approach that relies on the fact that the Fourier transform of a shifted signal has the same magnitude as the original signal, but with a linearly varying phase. So we used phase correlation to extract the two alignment parameters used in this study. The first parameter is the correlation coefficient R, which basically quantifies how good the resultant alignment was. And the second parameter is a translation vector, here denoted as P, that provides the transformation that we need to apply to one of the images of each pair basically to the correction that we need to apply to one of the images to maximize the alignment. And we use the tracking libraries of ImageJ and Fiji to calculate both of these parameters, R and T. So we had about 180,000 pairs of overlapping tiles for this acquisition, and for all of these, we calculated both alignment parameters, so R and P, the correlation coefficient and the translation vectors. So, and here we see the distribution of the correlation coefficient of R, and we see that most pairs obtain a very high correlation. However, there's a considerable portion of these that we didn't find an acceptable alignment. So if we depict these R values in space that here are seen on the left, so these are the correlations with high values are shown in warm colors, and the poor alignments are shown in cool colors. And we see that the vast majority of low quality alignments are concentrated in specific regions. So we want to know, okay, so what do these regions correspond to? In the right-hand side, we have a rough reconstruction of the data set, and we can see that the regions of low correlation values correspond to regions where a low signal was obtained. So just as a reminder, this is just an overview of a first successful map of the human hip. So it worked as a first feasibility study to develop, validate, and improve the methodology. So why did we obtain those low quality imaging? In some reasons, these had to do with the sample preparation process. Basically we had some of the sample regions had topographic depressions and cracks, and these were created by residual stresses in the microscopic samples, which were released during the sample preparation process. So such topographical abnormalities, they will promote surface charging of electron beams, and therefore will compromise the detection of secondary electrons. So these low signal regions were manifested either as darkened or blurred areas, and so these really highlight the challenges of preparing bone tissue for such large acquisition. So bone is literally a very hard material to prepare, and these highlight the challenges of preparing the bone tissue for methods designed to atomic force and electron microscopy. So we put a big effort in addressing these points, and the samples prepared with our new methods should not have a significant artifact. However, this was a robust test bed as a first feasibility study and as a validation for the technology. So going back to my point, so if we have regions where we had some topographic depressions that created low signal, and low signal led to regions of low correlation. But now we have these large collection of tiles, and what happens is that the tiles don't come up in pairs of overlapping tiles. They have multiple overlaps each. So therefore, the consecutive placement of tiles in the output image can lead to the propagation of alignment errors. And this is due to the fact that pairwise corrections may conflict when the position of a tile is defined by more than one neighbor. So we need to find a globally consistent set of alignment parameters that minimize the misregistration between all pairs of images. So to globally register the entire data set, we minimize the residual errors using mass spring damper system dynamics, so a relaxation approach. Each image tile was assigned to a point mass, and for each pair of overlapping images, the point masses of each image were connected by springs. So we then solve the system and allow it to relax and reach equilibrium. So we chose this approach as opposed to other more common approaches, such as least square minimization, as a way of reducing computer memory requirements while maintaining feasible computation times. So saving computational resources becomes quite important for large acquisition such as the one presented here. We use the Newton's second law to model the tile's motion. We use Hooke's law to calculate the forces exerted by the springs, and added a damping force to help the algorithm avoid overshooting and reach equilibrium. The thickness of the springs was proportional to the quality of the alignment, and pairs with high R values would have priority in the relaxation process. Also, our calculations were performed using MATLAB. So here we're going to see a video. I'm not sure if you guys will be able to see it well, but this is basically an animation that illustrates this spring, this relaxation approach. So let's consider a pair of overlapping images, I and J. So we have the Bachmann equation there that we're trying to align, and so each tile each tile of the pair had in its center had a spring connected. So springs that connected the centers of both overlapping images. We had the spring in the X and a spring in the Y directions, and these springs will try to move the tiles in order to maximize alignment. If the two images reach the position of perfect alignment, the springs will exert zero force. Right. So, but before stitching, we needed to correct the parameters of those problematic regions that we mentioned before. So we considered 4 criteria that took the value of R and the resulting overlapping conditions into consideration. So we first identified image pairings that had a correlation value below 0.5, alignments that would result in a lack of overlap, or translation vectors that would go beyond the initial overlap. And also we identified pairings that had excessively large initial residuals. So to correct the alignment parameters, we considered a removal scheme where we just remove the springs of the rejected pairs and any tiles that became unconnected from the system. This approach worked, however, within our maps had some gaps, and we also we developed another approach that obtained very similar alignments and avoid the patchy look of the removal scheme. So how did we correct for the rejected tile pairs? So we first corrected parameter R. So if a certain pair was considered non-acceptable, we would assign a spring with lower stiffness to that pair, and then to correct parameter P, so that's the translation, we tried to predict a likely pair of coordinates for that vector. We found that each type of overlap, as seen in the figure in the left, had a fairly predictive value of P, so the translation. And on the right here, we see the respective initial relative position between each type of pair overlap. So in the next figure, we have the same plot on the left, and on the right, we have the positions of all pairs I and J of images I and J only for pairings of high quality. So pretty much on the left, we have the relative positions of tiles before alignment, and on the right, we have the relative positions after alignment for the pairs with very, very high quality. And what we can see is that each type of overlap had a region where the alignment position was likely to be, so fairly predictable. So we used k-means clustering to identify the likely value of P. So because the value of R of these rejected pairings was low, these parameters in the end had little effect on the positions of the regions that had higher quality alignments. So here's another animation of that illustrates our relaxation algorithm. So here we have 5 hexagonal fields of view, and we have lines representing springs. And the line color here represents the respective local residual magnitudes. So green lines show the residual lengths that had good alignments, and the red correspond to high residual lengths, which we aim to correct. As it is noticeable, while the system relaxes, the high residuals or long residuals gradually decrease their magnitude until the system reaches a configuration where the vast majority of springs reach a satisfactory alignment. So this is a analogous animation. So in this video, we can visualize the repositioning of the actual tiles during the relaxation process. From here, it's hard to appreciate the quality of the alignment. So if we zoom into a cell on the bottom of that acquisition, I'm not sure how well you can see these videos, but we can see that while our algorithm solves, we see that most misalignments are reduced, and we reach a very, very interesting and acceptable alignment. Also, these videos are on YouTube, so when you get the PDF of these slides, you can see these videos maybe with a more quality and smoother transitions. So going back to our algorithm, or a relaxation algorithm, we had three mechanical parameters. We had the mass, the damping coefficient, and the spring constant. And we found something interesting was that the speed of computation was affected for these parameters. So we ran a parametric study where we varied the values of these parameters in a region with about 430 tiles. And we used the damping ratio as a non-dimensional measure of the mechanical parameters. And we can see that there's a range of damping ratio values that for which the system solves in shorter amounts of time. So before running our stitching algorithm, we ran a parametric study, found an optimal damping ratio, and then we extracted our mechanical parameters using such parametric study. All right, so as I mentioned before, we had the option of using least squares minimization to globally register the whole data set. So now let's compare the performance of our algorithm to least squares minimization. We used basically a region of 60 multi-beam fields of view. So, and we can see it in red in the figure in the left-hand side. And we calculated the root mean squares of the residuals for our approach and for least squares method. So when we calculated stitching using both approaches. And we see that the initial residuals that started around 47 pixels were reduced to about 7 pixels for both approaches. And the final tile configuration of both approaches also very close. So this demonstrated the capability of our method. We also perform all calculations in MATLAB here. And as I mentioned before, the reason why we chose a relaxation approach over least squares minimization was that we wanted to reduce RAM usage while maintaining feasible computation times. So our approach took roughly 10% more time than least squares method. However, it used 10 times less RAM memory seen here on green, which is pivotal for acquisitions as large as ours. So I've been talking about, I've been showing results of some smaller sections, but what about the, what about the whole collection of almost 55,000 images? So here we see the length of the residuals for all pairs of tiles. And initially we see that most configurations are misaligned by more than 10 pixels. So, and during our algorithm calculations, we see that these values are reduced to the 1 to 5 pixel range. Now, if we look at this from a spatial point of view, so in space, we see that the local residuals are how they are reduced during the course of our simulation. So here, the high residual values are shown in yellow and green, and they transition to cooler colors representing lower values. Yeah. So this, and after this process, we obtain the final positions of our. So after the stitching process, we wanted to make these data sets available to the community and public alike. So we used the Google Maps API due to its familiarity and ease of use to harness the potential of these maps. So we started by pre-rendering the various zoom levels of our data sets and created a tile pyramid structure
to be used with the Google Maps API. So in our website, MechBio.org, you can test one of these maps that were acquired using MSEM technology. So MechBio.org is our lab website, and we aim to soon make it a reference library of maps of a variety of human patients. And we are working in a robust and reproducible pipeline that will be applicable to all types of MSEM datasets. So here's just a video illustrating how the map works. We see, I don't know if you can see it well, but we're zooming into a region where we can see an aversion system with a blood vessel. Here we're zooming into a cell, an osteocyte. We can see the dendrites and how the cell is embedded in the bone matrix. Yeah, we can see very well the morphological details of the tissue and also of the biological components. And zooming here into this blood vessel here, we can see a very nice picture of an osteocyte connected to the vascular space. So now I will pass to my colleague Dan, which will show some of the applications of such platform in our research. Thank you.
Hi, everyone. So I'm just going to talk to you all a little bit briefly about the applications that we now have using this new geonavigational platform. So as I'm sure all of you are actually quite aware of Google Maps and its uses, whether it be at the local scale looking for the nearest grocery store, or perhaps looking at the various bus routes to the closest scientific center to attend your conference. A lot of us have used Google Maps for various issues and have relied on it in our day-to-day life. So it's really key that this platform that we've chosen is very well known and understood to both the scientific and public communities alike. So likewise, by using this platform for our work, we similarly can look for sites of interest and drop pins on what we identify to be such features of interest that we may later on want to analyze. So if we look here in the top right, we have that cell that André just showed you, which we mark as a healthy or viable osteocyte. As you can see, it has several processes extending from the cell body. We defined a viable osteocyte as having at least 3 or more of these processes. In the bottom left, we can see what we consider an unhealthy or pygnotic osteocyte, which is marked in red, having less than 3 extending processes. Then here in the bottom right, we actually outlined the lumen of a blood vessel, which is part of the vasculature, which we find in many of our samples. So if we quickly go back now, we can look at the video of how we mark these maps. So right here, we have this map that André just showed you, and now we can zoom in to individual cells. Here we begin to mark the healthy osteocytes. As you can see, we can navigate around quite easily. We find the cells that have many processes sticking out, and then we mark them in their respective green color. And then as we begin to zoom out, we can pan over to the right, and we see what looks like an unhealthy osteocyte or pygnotic osteocyte, which we mark in red because it doesn't actually have any processes. Scrolling over to the left, we find a blood vessel where we mark the outline of the blood vessel borders in blue. As we scroll up, we actually come across a viable osteocyte around the border of that vessel, which is actually where your typical new osteocytes will develop and originate. So what we have now as we zoom out is you can see we have hundreds to even thousands of cells and vasculature markings on this map, which gives us basically a lot of data from one sample, which we can then begin to analyze. So with this newly marked map that utilized the Google Maps platform, we can begin to look at the various epidemiological studies across populations. Now, the same way in which you might run a study across a population of a certain country, we can now investigate populations of specific cells within our specified tissues. So, for example, by merely assigning coordinates to the designated cells and vasculature, we can begin to develop network models that allow for a thorough characterization of the specimen under investigation. And this is a model that would lead to comprehensive diagnostic insights that we're able to compare both healthy and unhealthy tissue across physiological networks. So in our case, just investigating one sample via our Google Maps platform, we've begun to just briefly look at the example of the mean path distance of both viable and pyknotic osteocytes and their relationship and distances to the vasculature and specific blood vessels. So as our sample sizes increase, we will be able to cross-correlate amongst samples and eventually amongst individual patients, creating network models, and this will enable great strides within the realm of pathological diagnostics. So with that, I will hand the reins back over to Melissa so she can bring this all back together.
Thanks, Dan. Thanks, Andre, for your beautiful work. It's a real joy to work with such a talented team. So as Andre outlined, the methodologies required to build a stable pipeline, essentially, for handling these data sets, building the maps, and Dan outlined how we then can use those maps to start to make important physiological both discoveries or to test hypotheses. Our ultimate goal is to recognize changes in these cellular networks at very early stages, so we can either stop the degradation of the network or we can reverse the process. And it turns out as we age, many of these degradation processes, they're a natural part of aging, but in many diseases they're accelerated or they reach such a profound level of disconnect that function is compromised, such as in dementia or in osteoporosis. So we're very much looking forward to the development of next generation diagnostic capability where our understanding of these the connectivity of these cells, whether they be bone cells or brain cells, goes from beyond 160 micron on edge sample size or volume of interest and goes to a whole organ. Because as you can imagine, we could pick up individual volumes of interest with previous imaging methodologies, but then you have a real issue of sampling, choosing the correct sampling points. Using these seamless multiscale modeling methods that are enabled through the MSEM technology, we can now see across the whole tissue, across the whole organ to pick up those small areas at very early stages. And so we can use, similar to before, we can just expand these models to include much, much larger data sets. So ultimately, we want to predict the earliest stages of disease emergence. I hope that through today's webinar, we've been able to convince you of what we recognize as an incredible power of coupling multiscale imaging and modeling to understand incipient disease processes. And in addition to understanding the incipient disease processes, we see these enabling technologies as a pathway to using transdisciplinary approaches, both across tissues and organs, but also to develop next-generation materials, next-generation implants, and diagnostic devices. And this has been highlighted. We have a perspective article out recently in Advanced Healthcare Materials highlighting these shared paradigms between different tissues, different organs, and hopefully we're going to start to understand the processes that happen within individuals, within those cellular populations, relate between individuals, and then even at a larger length scale between different populations across the globe. I would like to thank my team very much. I'd like to acknowledge Eric Anderson, who I mentioned earlier is now the Chief Fluids Dynamicist at NOAA. He started this work with paracellular, nano to micro fluid dynamics approaches to understand structure and function and the interface of mechanics and cellular biology, as well as my team here in Sydney, in particular André Pereira, who presented his beautiful work on creating a pipeline to create these maps, to validate these maps, and to use them in a standardized way going forward as we collect data from a number of human patients. I'd like to also thank Dan Hagema, who presented his work on geonavigation of these maps and how we can use these for next generation network modeling. I'd also like to thank Dr. Ulf Knothe, our orthopedic surgeon colleague, who in collaboration with Cleveland Clinic under an institution review board protocol, we were able to obtain these specimens from human patients who were undergoing hip replacement. So these were tissues that would normally be discarded during the normal course of the operation, and this was a really important step, as well as Zeiss microscopy, in particular Dr. Dirk Tate, with whom I've worked the past 5 years, and it's been a wonderful collaboration with Zeiss, seeing this incredible technology, the MSEM, go from prototype to a commercialization stage. As I mentioned, if you have any questions, please do refer to our publications, in particular the PLOS computational biology paper that should be coming out any time now. This is our web portal. Please do contact me. We're recruiting PhD students and postdocs at all times, so we're always interested in new talent. And we'd also very much like to thank our funding agencies without whom this work would not be possible. We look forward to having a discussion with you, addressing any questions you might have. Very interactive discussion, so we can go back to any slides you might have questions about. And André and Dan are here with me, so we're happy to address and discuss. Thanks so much for your interest.
Thank you very much, Melissa. I thought that was a really fantastic presentation, and I think this is really cool that you have this integrated with API, Google Maps, you know, and I actually was playing around with it while I was watching the presentation as well.
Great.
I had, so do you kind of envision, like what would be, so you generate these kind of maps, they're probably going to inspire other people to generate these kind of maps. Is there some kind of repository, some kind of centralized place that you're envisioning having not just your own stuff, but like user-contributed type of content?
Yes, definitely. So as mentioned before, we are developing the MechBio portal so that our maps from human patients become available to scientists as well as the public. And so I think public engagement is really important in the work that we do, since the public pays the taxes that pay for the grants that enable the research. So we have an obligation to the public to show that our work is relevant and important, and we hope to engage the next generation of scientists and engineers as well by getting school kids involved. Our colleagues, Professor Lichtman at Harvard and Professor Dank in Germany, have been working on this brain connectomics initiative very hard, and they've been real pioneers in the development of the brain connectomics initiative, which was also with Professor Sebastian Sung at Princeton. They have been working primarily in mice, and they also have repositories for their images for mice. This, in my opinion, the imaging and the computational modeling are taking off at such a rapid rate. It's the efforts of IMAG and groups like IMAG to make these available to the public, as well as to scientists, so that we can move the science and the discoveries forward as quickly as possible. And without dominating the discussion here, I should also mention these are huge data sets. So that's one of the challenges of doing this coupled imaging and modeling, is having a place where you can have them readily available and access to such huge, you know, these are terabytes of information per map. So that becomes a major challenge. And the field is, it's really interesting, the field is rising to this challenge, I have to say. When we first had some of these data sets, also with some other imaging modalities, we had even trouble publishing them because we were having trouble with the review process. How can you upload to an electronic submission site for a journal a terabyte of data? You just can't do that. And so we're working with journals that are very interested to be at the vanguard and to enable rapid dissemination of this information. And so the journals themselves have to, and the publishers have to come up with ways for us to do this.
I have a couple of technical questions about SEM, but I would like to see if anyone else that's tuned in here has any questions while we're still on this topic.
I had a follow-up question to Ed's question in terms of the repository that you're hoping to set up. So do you envision the interaction to be on your server or in the cloud, or how would people actually be able to utilize your data then?
At the moment, it's through a physical server here at UNSW. Our Faculty of Engineering has been very generous in basically providing this for us and providing the support. Ultimately, we're going to need to have this on the cloud. We're hoping that Google or some other industry partners may come up with a donation or some sort of public access possibility so that we can make these very accessible for the public.
I wonder if Google itself might be interested in helping out with the storage and the maintenance of such kind of data sets.
We definitely hope so. We were very honored to be invited to Google to give a Google Tech Talk. So I gave a talk in July of last year. Ironically, Google Maps, I don't know if people are aware of it, but it originated here in Sydney. And so we have the best Google Maps capability in Sydney, apparently in the whole world, where we can essentially go and Google Maps anything, and we can find how to get there by foot, by car, and by public transport. I think that has disseminated much through the world, but we've had that capacity for a long time due to the proximity here. So they've been really very interested, and we're hoping that they'll up the ante to getting this information out.
So I want to ask a couple of technical questions about the SEM, only because when I was a postdoc, I spent a long time acquiring images in a dark basement with an SEM, and I only had one image at a time I could take, so I would manually trace across, my samples and collect, what seemed ridiculous to me, 60 or so images, and then try to piece them together myself. So, and to me, the sheer quantity and the fact that you've got this array that allows you to get, 64 images or something like that at a time. One of the technical questions I had was, this is a very detailed question. When you have an array set up like that, do you have charging effects that happen differentially on one image of the array versus another that causes things to, so you have electrons that are ruining the image and creating artifacts in one component of the array, or is that a real challenge, or it doesn't really present itself as an issue?
Well, let me just, the prototype that this map was created on was a 61 beam prototype. This is now commercially available, and you can even get one of these MSEMs with a 90 beam. So the number of beams is going up and up. In this specimen, the charging effects are evident due to the problems with the sample preparation, because it was literally a first in the world. These sample preparations are now much less problematic because we're able to prepare the sample in a way that we don't get these sorts of literal hard artifacts where the electrons charge up. Also, I should note that Dr. Dirk Zeidler, I think, is in on the webinar. So, Dirk, I don't know if you want to make any comment about the charging effects and the challenges.
So, can you hear me?
Yes.
Okay. So this is Dirk Zeidler speaking. We have to ensure that the samples are well conducting such that charging effects are minimized, actually. But let's say this is something we're definitely working on.
Great.
Okay.
Thank you, dear.
The charging on samples was like the bane of my existence, so I had to ask the question.
Well, and you know, just to make a comment, these kinds of images, these maps, they would, with the techniques that you used in the dark basement, they would take decades to produce back with that technology. And to think that we can do this so rapidly now is just really mind-boggling. So you know, and so there's both the technology of the imaging as well as, you know, you've got this millimeter on edge specimen back in the days of working in a dark basement, where now we've got these real-sized human hips. So these are literally much larger specimens, and so it's the technology, the imaging technology, but then also how we prepare these specimens, and that's not trivial, because if you're preparing something a millimeter on edge, you can get it perfect, but we're getting there. And the other interesting thing, I think, is the etching protocols. I don't think we talked about them very much, but so we get this perfectly planar surface, and then we can use atomic force microscopy protocols we developed way back in the days when I was a grad student in Zurich, where we specifically etch away the mineral or the organic component, so we can really start to pick up the different components of this large structure and this sort of resolution. And you can't see it here, but in the best resolution of this device, we can see down to individual collagen molecules, collagen fibrils. It's really mind-boggling.
Yeah, so, you know, one question that comes up here, because it's a question I've had with my own SEM images and stuff, is, you know, you get this exquisite detail and resolution, but everything you do to prepare the sample or take it out of its in situ environment has the potential to create artifacts. So in some sense, you can zoom in so that you kind of lose fidelity or something like that. And I'm sure you've been thinking about that issue. I'd be really curious to hear kind of your philosophical musings on that.
Yeah, so we do a lot of this, and that's always our concern. And ultimately, I hope within my lifetime to be able to do this kind of imaging live in the patient. Obviously, we won't be able to use this particular technology, but imaging technology is moving forward so quickly. If I think about how we're able to watch the dynamics of cytoskeletal proteins adapting to mechanical stimuli in stem cells, in live stem cells, so that's coming from really that bottom up. And now we're going with this top down. I think we're going to reach that at some point. In these particular specimens, we have a lot of experience. You know, my career lifetime of experience of sample preparation, where we started in Zurich with looking at these on the atomic force microscope. So there, any changes, any artifacts are amplified even that much, right? So we try our best to keep the sample intact, and then we etch away from the surfaces, so we look below the surface, hopefully at the most high fidelity representation as possible.
Okay, well, does anyone else have questions?
This was a fantastic talk, Melissa. Let's give Melissa a round of applause virtually.
Thanks to my team, and I'll put up this paper once more. Please, for the full details, I know it went pretty fast, so the full details are in this paper. Please check out the maps, and there'll be more coming out, so we'll keep everyone posted. And we look forward. I saw there's the 10th anniversary IMAG celebration in March of 2017.
Yeah, so we can, if you can post this reference on the webinar or on your working group